Now to the main business tonight, which is tonight's lecture. I'm absolutely delighted to invite Linda Partridge to give us a lecture this evening. Linda is an internationally known researcher who works on the biology of aging, and in particular, healthy aging, which is, of course, a big societal concern. I won't say anything about it being a concern to those of us in the audience. Linda started her research career studying the behavior of birds, and she then moved on quite quickly to start studying aging. She was at the University of Edinburgh, and then she moved to University College London, where she became the Weldon Professor. She was also the founding director of the Max Planck Institute for the Evolutionary Biology of Aging in Cologne. Now, Linda's had lots of awards and honors for her research. I'm not going to run through them. She's also a fellow of the Royal Society. She's a fellow of the Academy of Medical Sciences and a fellow of the American Academy of Arts and Sciences. And she is a dame of the British Empire, and she's cringing at that. Please, Linda's title of Linda's talk is Healthy Aging, the Hype and the Hope. Thank you, Linda.
Thank you very much, Pat, for a nice introduction. And, you know, it's a real honor to give a lecture to the Royal Philosophical Society. Thank you very, very much for inviting me. So I thought I'd start with Billy Connolly, famous Glaswegian, of course, because he really covers the two sides of what I'm going to be talking about. Because in many ways, he's a very healthy ager. He's 83, he's enjoying life in Florida, he's a very active artist. But on the other hand, he had a brush with prostate cancer a few years ago, and for many years, he's been dealing with Parkinson's disease. So both of those have lowered the quality of the later part of his life, two classic age-related diseases. So the sort of thing that I do, and many others, including one or two people in this room, I notice, is to think about how we can push back the age of onset of the bad things that happen with age, age-related diseases, but also more general loss of function. Can we somehow squish that period of ill health at the end of life? So that's the name of the game. And we've certainly been very good at making ourselves live longer. So this is by now a classic illustration of the historical trend. So it started way back in the middle of the 19th century and has sort of continued to the present day. So what you've got there is life expectancy at birth. So that's the length of time that the average person is expected to live against the year in which the birth took place. And it's plotted for the country that was the world leader at the time. So to start with, that was mainly the Scandinavian countries. Latterly, it's been Japan, South Korea, the East Asian countries. And the causes have been different at different times. To start with, a lot of it was to do with infant mortality, infectious diseases, antibiotics, immunization, and so on. But latterly, the increase has been mainly in the older section of the population, basically because of much better medical care, as well as better living circumstances. The horizontal lines are just the predictions of various individuals and organizations about where the trend would top out, what the intrinsic limit on human life expectancy would turn out to be. And it basically shot past all of them. That was the point of the publication at the time. So it's 2.5 years per decade, the slope of that line, 6 hours a day. So quite extraordinary increase in life expectancy over that period. And of course, it's led to some extraordinary lifespans. I think these two are interesting. They're always women. On the left is the, as far as we know, the world record holder. So she was a French lady. There was a suspicion for a while that something had happened there that we know does happen in other cultures, which was that her death had been concealed, and it was in fact her daughter collecting her pension. But I think that one's been quashed now, although certainly some, for example, the idea that people in Okinawa in Japan live a very long time seems to have been attributable entirely to that phenomenon. But hers seems to be a genuine record. So she made 122 and a half. And the lady on the right's the oldest living person at the moment. She's actually English. She was an au pair in Hong Kong, and she attributed her long life to never getting into arguments with anyone. She says, I just listen, and then I do what I want. And also having an optimistic attitude. She actually survived COVID when she was 110. So really quite remarkable. Cologne, I think, is even more mysterious with that great record. She smoked until she was 117. So, and that's quite true of these extraordinary centenarians and supercentenarians. It's not obvious from their lifestyle why it's them. But clearly this is, really something to celebrate. We've improved our living circumstances over this very long period. So we're staying healthier at a given age and we're living longer. Very positive feature of civilization. But if we look a bit more closely, there are some wrinkles and granularity makes the picture a bit less pretty. So I've been looking for some of the statistics in Scotland. And these are the very recent figures for life expectancy. This is from Scottish Government records. So this is between 2000 and 2022 on the right. Again, it's life expectancy for women and for men. So the women in yellow, the higher line. So everywhere, women live longer than men do. That's a global phenomenon, but they're not healthy extra years. So women have a longer period of ill health and loss of function at the end of life than men do generally. And also, if you look at the trend here, what you can see is that the early part, up until about 2010, you see the increase continuing, but then it plateaus before COVID, and it hasn't recovered. And if anything, it's actually coming down again now in both sexes. And also healthy life expectancy in Scotland has actually declined since 2014. So that's defined as the age at which the first age-related disease occurs. So it's the age-related disease-free period of life. And that has decreased steadily since 2014. So what's happened as lifespan has increased is really illustrated by these figures, which is, although we're living longer, actually that period of ill health at end of life is getting longer as well. And the big picture here, of course, is one of degrees of social deprivation. It's really socioeconomic, this kind of trend. So again, these are figures for Scotland. And what's shown here is life expectancy for the most, second most, and second least and least deprived areas of Scotland. This is actually rather coarse. I'll show you in a minute. These are quite large areas. But what you can see is the very clear relationship, something like 5 or 6 years of life expectancy difference between the most socially deprived and least socially deprived areas of the country. This was how they actually cut the cake into different areas. If you actually look more fine grains, this is typical in London as well, over very short distances, you can get huge variation in the life expectancy of the people living there. So this is really one of the huge things that needs to be tackled if we want to improve people's health as they age. But however you look at it, we're living much, much longer than we did in our evolutionary past. And that means that natural selection hasn't had a chance to mold the later parts of life. We've basically evolved to function well when we're young, but not as we get older, because natural selection hasn't had a chance to modulate things later in life. And that's why we see this late life loss of function. It's because of the huge increase in life expectancy. And what that means is that we're starting to see this incidence, steady increasing incidence of age-related diseases. And I can illustrate that with some work that was done recently at UCL. This is actually looking at National Health Service records, primary and secondary care, so hospital admissions as well as GPs. And the two people who did this, Val Cohen, extraordinary person. She did a degree in maths. She then became an actuary, got bored with that, so became a trader on the stock market floor. Decided she didn't much like that, so she trained as a medic and became a GP. And by the time I got to know her, she was doing a PhD on these National Health Service data. She's actually now a GP in New Zealand. So extraordinary life story. But she analysed 3 million national health records. And what she asked was which diseases, how many diseases, are age-related in their instance. So she looked at these 278 high-burden diseases, ones where a lot of people get them, and simply looked at the rate of incidence and how it changed with age. So incidence is the first occurrence in someone's life of the disease. And she came up with these 9 clusters. So when she looked at the incidence rates, they fell quite naturally into these 9 different patterns. And you can see that the first 4 of them, the 3 at the top, and then the left-hand one in the next row, they're all age-related. So basically, they're progressively younger incidence as you go through the 4 groups, but all of them go up in incidence with age. So the first group, which really only comes on very late, hardly has any diseases in it. So delirium, hip fracture are in there. Cluster 2 actually is mainly cardiovascular disease, pretty late onset generally. Cluster 3, most cancers are in cluster 3. And 4 is a bit heterogeneous, but there are a lot of digestive disorders in there, for instance. And those 3 between them account for 207 diseases. So 207 of these 278 are age-related incidents. So advancing age really is the major risk factor for most diseases. And what that also means is that you start to see a lot of people who've got more than one disease, which is called multimorbidity. These are actually data for Scotland because they illustrate it particularly well. These are data for men. Women look pretty similar, and it's a pretty general picture for other places. So again, as you go through advancing ages, what you see is that, so for instance, by the time you get to 70, most people have got at least 3 diseases, and almost nobody is disease-free at that age. And multimorbidity is complicated because often there are conflicting requirements for treating different conditions. And also you start to see interactions between drugs and so on. So it's a real challenge often to treat multimorbidity. So what this means is that the major burden of ill health in our society is falling on the older section of the population. And there really is a global challenge to try to do something about it. And what we would like to do, as I've mentioned, we've seen this big increase in lifespan. We've got the unhealthy period at the end of life. And as we've increased lifespan, we've actually also increased the unhealthy period. So health span has increased, but not as much as lifespan. And we're seeing this longer period of ill health. But what we'd like to do is this. We'd like to have the increased lifespan, but with a squish of that red period at the end of life. So is that even a realistic goal? I think it's important to realize how malleable aging actually is. So there are some creatures that don't age at all. These are 3 of them. We've got a sea anemone on the left, little freshwater hydra in the middle, and a planarian worm on the right. And as far as anyone can see, they don't get any more likely to die or any less able to reproduce as they go through their lives. They just flatline the whole way through. And an interesting thing about them is that they are more or less bags of stem cells, these dividing cells that can regenerate. They have great powers of regeneration, these animals. And the stem cells can also give rise to the germline, the sperm and the eggs, when they reproduce. So they've got an evolutionary future. And this may be the key to why these things don't age at all. But they're a clear existence proof that aging doesn't have to happen. And also, if we look more broadly out there in nature, there are some amazing lifespans. that seem to be independent of other characteristics. Aging seems to be a feature that can evolve to different values. So I think the bowhead whale is the mammalian record holder at 200 years. Of course, it's very big. So you might say, well, of course it's long-lived because it's big. But very small things can live a long time too. So if you look at the bottom row, the ocean quahog, little bivalve mollusk, it can make 500 years. Naked mole rat is a rodent, very interesting social life. It's about intermediate in size between a mouse and a rat. They can live 30 years, whereas a mouse can only live 3. And this little Brandt's bat I find the most amazing. You can see it's just sitting on the tip of someone's finger. It only weighs 8 grams, and it can live nearly 40 years. So you've got aging here evolving quite independently of body size. I just put the Greenland shark up because it's such a crazy creature. It lives quite deep in the Atlantic. It can live for 400 years, and it doesn't even start reproducing until it's 150. So it wouldn't make a great fishery. But clearly you've got aging, the rate of aging evolving out there quite independently of other characteristics. And these differences are genetic. Somehow, these different rates of aging are being encoded in the genomes of these different creatures. So an obvious way into looking at what we can do about aging is to think about the genetics. Can we find some of the genes that actually influence the rate of aging? There are some genes in humans that do the opposite. These are the premature aging syndromes. Mercifully, they're extremely rare, but they do actually tell us something about mechanisms of aging. So these are the 2 main ones. Hutchinson-Gilford progeria on the left is the most extreme. So the children are born ostensibly completely normal, but at the age of about 18 months or 2 years, they start to show the symptoms. You start to see loss of hair, loss of subcutaneous fat. Those are the obvious superficial things. And they generally go through to about 13 years of age. And as I say, mercifully, it is extremely rare. Werner's syndrome is less extreme. It has an onset in the teenage years. And usually they make about 50 years of age. And the Hutchinson-Gilford children get cardiovascular disease. Werner's syndrome get both cardiovascular disease and cancer. Those are the main causes of death. And eventually the single genes that were mutated in both of these diseases were identified. And interestingly, they're both to do with maintaining the fidelity of the genetic material. So Hutchinson-Gilford, the gene actually encodes a protein that's present in the nuclear envelope, and it's very important for maintaining the structure of the nucleus and the arrangement of the genome in the nucleus. And that seems to be what goes wrong in that syndrome. And in Werner's syndrome, it's actually something called a DNA helicase. It's a molecule that's involved in replicating the genetic material, and when it's mutated, there are mistakes in the way that the DNA is copied. So I think that's telling us that it's very important to maintain the fidelity of the genetic material if you want to age healthily. But of course, ideally, what we'd like to find is mutations that improve health during aging, not that make it worse. And for that, really, this cast of characters have been the key. So these are the laboratory model organisms, I'm sure familiar to many of you, but we're talking about yeast, the nematode worm at the top right, Caenorhabditis elegans, my own favorite organism, Drosophila, down in the bottom left, and also the mouse. And they've really been engines of discovery of mechanisms in biology, things like how the genetic material is replicated, how it's expressed, how the nervous system works, how metabolism works. They've all been discovered using these organisms. And it works because of the very strong evolutionary conservation of this kind of biology across big evolutionary distances. But I think people thought, well, is that going to work for aging? These things live very different lengths of time from each other. The worm lives about 3 weeks, fly about 3 months, mouse about 3 years. And they face very different kinds of challenges as they go through their lives. Fortunately, all those worries turned out to be unnecessary, and the track really started here with work on the worm, Caenorhabditis elegans. So this was a very famous experiment by Cynthia Kenyon, which really kicked off a lot of the modern science of aging. And what she did was a very simple mutagenesis experiment, and she said, can I isolate a genetic strain of worms that lives longer than the normal wild-type worm? And you can see it here with the black dot. So this is a survival curve now. We're looking at the number of worms alive as they go through their lives at different ages. And you can see that the mutants living much, much longer than the wild type. And very importantly, they were still wriggling around and healthy looking long after the controls were dead. It certainly was not just an extension of the moribund period at the end of life. So that was in itself quite a dramatic finding at the time, but it took quite a long time to figure out what this actually was. And it turned out to be a mutation in the single worm insulin-like growth factor receptor. So this is a molecule that sits in the membranes of the cells and responds to insulin or to growth factors. At the time, much better known in mammals, insulin because of the whole role in diabetes, insulin-like growth factor because of the role in growth and wound healing. And I think nobody even suspected it would be present in worms at all. But it turns out that just mutating, tamping down the activity of this receptor improves the health of the worms during aging and makes them live longer. And quite interestingly also, there are a number of worm genetic models of human age-related diseases, so neurodegeneration, cancer, and so on. And it turns out that these mutants, if you combine them with the disease mutants, can often rescue a lot of the pathology. So this was one experiment that was done with C. elegans. So this is actually an Alzheimer's disease worm. So it's expressing one of the proteins that's important in Alzheimer's disease, but in the body wall muscles. And you can see that it induces a sort of paralysis. The whole of the back end is paralyzed, and there's just this rather feeble moving around at the front end. But if you combine that now with that DAF2 mutant that I've just shown you, you get a nice healthy worm again. And this is also true with worm models of cancer. So this mutant's really powerful. It can improve the health of the wild-type worm and rescue this disease pathology. So just what the world needs, a really healthy worm. But happily, it turned out to be evolutionarily conserved. So first of all, people looked at Drosophila when the whole pathway came to light there, and then eventually in the mouse. So these are mutants in the fly and the mouse that are very closely related to that worm mutant. And you can see the nice extension of lifespan. And just as in the worm, they could often rescue disease-related pathology. And in the mouse, there was a very thorough look at the phenotyping. And I think what was really surprising about this story was the very broad spectrum improvement in health in different systems. So they had better glucose homeostasis, better immune profile. I should say the person who did this work sitting in the room, Colin Selman, and better motor function. They maintained their coordination better with age. They got less osteoporosis and cataract. And you can see the ulcerative dermatitis on the right-hand mouse. That's the control mouse. About 40% of them got it as they went through aging. The mutants were completely rescued. So I think what's really interesting here is the broad spectrum improvement. And it seems that what's happening here is that exactly what I've said about us functioning well in youth. The same is true with these animals. They're laboratory model organisms. They're living in a protected environment. They're living much longer than they do in the wild. And it turns out that this signaling system, which is what it is, works fine when they're young, but it drives too strongly at older ages when other things have started to go wrong. So if you tap down its activity, it helps. The way that this has been followed up in humans is just by looking at natural human genetic variants. So you can look at the human equivalents of these interesting animal genes, look for variant sequences in them, and ask if they're associated to survival or to great ages in humans. And there's been quite a bit of that, and it really supports the idea that this whole mechanism is probably also present in human aging. So these are just various papers that came out on it over the years. You can see some of the titles. These are all bits of this signaling network. FOXO3A, the top left one, is very interesting because this is a key, it's called a transcription factor. It's a molecule that turns on and off the expression of genes. In the DAF2 mutant, when you knock down the activity of the receptor, it's activated. And it turns on the expression of genes that are involved in defense and stress resistance and so on. So in a sense, what these mutants are doing is tricking the animal or the human into thinking there's a problem, there's a shortage of nutrients, or there's a stress, when actually everything is okay. And that improves health later in life. So this is just one of the whole mechanisms of aging that have come out in that short period since the early 90s. But there's been a huge burst of other work in parallel. And that's come up with these hallmarks of aging. These are the basic mechanisms of aging that their contribution varies between different tissues and cell types, and to some extent between different types of organisms. But they're there in the aging process, and they've all been shown to be causal in producing the problems. So you could actually show that interfering with them or modifying them in some way improves some aspect of health during aging. So I think everyone thinks it probably kicks off with things going wrong with the genetic material, as I've already mentioned, the top three, so mutations, nibbling away at the ends of chromosomes, the molecules that stick on to the DNA going wrong. And that means that cells gradually lose control of their function. So the important effectors in cells are the proteins. That whole domain starts to go down. There are mistakes in their sequences. They form aggregates and so on. I've talked about the nutrient sensing with insulin. The powerhouses of the cells, the mitochondria, start to malfunction. You get this phenomenon called cell senescence. Basically, when DNA damage happens in cells, they often stop dividing, they don't die, they sit there, and they spit out damaging molecules into the rest of the tissue, and sometimes at a distance. So they cause a problem. Stem cells don't do what they should do. They don't divide when they should, or they misdivide when they shouldn't. And you also get a lot going on at the whole system level, particularly immunologically. There's a major increase in inflammation during aging, which is very common. And also the immune system stops functioning as well as it should. And they aren't independent, these mechanisms, but they interact with each other. It's a complicated process. But I think the real achievement of the last very few decades has been to understand these fundamental mechanisms. And a lot of interest is therefore being directed now into saying, well, can we interfere with them to improve human health during aging? So just a couple of examples of very recent work. Muscle weakness, sarcopenia, is a very important loss of quality of life during aging. You know, it very often leads to falls and just general lack of mobility. And there's a very nice piece of work going on at the moment from the lab of Helen Blau in the US. She's discovered an enzyme that can restore the way that the nerves communicate with the muscles, the neuromuscular junction, and also the muscle strength and size. She's done this experimentally in mice. And that's basically acting through three of these mechanisms of aging, particularly important stem cells and mitochondria. So you can see on the right that the neuromuscular junctions, which are the yellow, they're beautifully arborized and restored by this enzyme. And she's now in phase 2 clinical trials in humans with this enzyme. So I think that's very promising. Something else that regularly goes wrong during aging is intestinal dysfunction. The structure of the gut goes down. It also becomes leaky, and that may be an important source of age-related inflammation. And there's a drug called rapamycin, which I'll say more about later, which induces a cellular cleanup process called autophagy. And that turns out to be able to restore both the structure and the function of the gut. And it's also looking as though it's really important in reducing age-related inflammation in humans. So, and there are a lot of trials going on with that drug. So I think these basic mechanisms of aging are now starting to feed through into looking at how we can interfere with them. And there's an even broader idea out there now, which is generally known as the geroscience hypothesis. Because those underlying mechanisms of aging are present in the etiology of age-related diseases, often several age-related diseases, we might be able to interfere in just one of these mechanisms of aging to prevent multiple things that go wrong later on. So the emphasis is on prevention and one into many. So as a sort of parody of current very successful medical approach, we tend to go organ by organ, disease by disease, medical speciality by medical speciality. But maybe geriatrics could develop more into a science where we actually interfere with the mechanisms of aging to prevent diseases in several systems simultaneously, as in that mutant mouse earlier on. So could that idea apply to humans? Is there any evidence at all that it might work? And it's something that we've been doing a bit of work on recently, mainly using data from the literature and from patients. So these two were the ones who led the charge here. Helen's a PhD student in the lab with Madalena, who specializes in computerized reading of the literature. And what they did was to machine read almost 2 million abstracts in the biomedical literature, so that vast literature out there. And what they did was to look for mentions of those 207 age-related diseases, which I talked about earlier from the work of Cologne, and a mention of the 9 aging hallmarks. So they were looking for a co-occurrence in the abstract of a mention of the mechanism of aging and a mention of the disease. And that let them do for each of the 9 aging hallmarks to produce a list of the 30 diseases that were most commonly mentioned in association with that hallmark. And this is what that looked like when summarized. It turned out to be actually only 83 diseases, because many of the hallmarks were present in several diseases. And in the boxes here, what you have is the strength of the association between the hallmark and the disease. So dark red means mentioned together a lot. Yellow means sometimes mentioned. White means not mentioned together at all. And it's all very non-random, and it made a lot of sense. So for instance, mitochondria cropped up a lot in the context of neurodegenerative disease. And we know that that's the case. So they seem to be picking up a good, strong signal. So we've now got 30 diseases that are associated with a hallmark, say stem cell exhaustion. We can actually go back and look at those medical records, the National Health Service records, and say, if diseases are associated with a single aging hallmark, are they also more likely to occur together in patients in multimorbidity? And the answer was yes, they are for several of the hallmarks, quite remarkably. So we've got these age-related diseases from Cologne. We take 4 decadal networks in the patient. So we start at age 50, late middle age, and look at 50 to 60, 60 to 70, and so on. And we can make a network of the 30 diseases associated with the hallmark. So this is shown here for the 30 diseases associated with stem cell exhaustion. So what you've got again is the color coding, the strength of the association with the hallmark, and the thickness of the black line is the extent to which the diseases co-occur in patients, the pairs of diseases. So a thick black line means those two commonly occur together in the same patient. And we can say, well, are these networks for the diseases associated with a hallmark stronger than we would expect by chance, given the structure of the overall network? I mean, clearly there's a lot of statistics in there, but it can be done. And it turned out that for 5 of the hallmarks, so nutrient sensing, mitochondria, cellular senescence, stem cell exhaustion, and altered intercellular communication, the densities were higher. So what that's telling us is that there are some patients who have a predominance of mitochondrial dysfunction, and so they are getting the age-related diseases associated with mitochondrial dysfunction. They're getting them together in multimorbidity. So therefore, if we could tackle the mitochondrial dysfunction, we would take that group of diseases down simultaneously. So I think there's real promise here that we may be able to take this kind of approach to age-related diseases. And of course, I mentioned at the beginning the real importance of socioeconomic situations. And it seems that quite a lot of the variation between individuals here, in whether they get particular sets of hallmark diseases or not, is associated with social deprivation. Again, this is something that one can do particularly at UCL because so many of the longitudinal cohorts, and including UK Biobank, are present there. So for this, we collaborated with Mika, who's the professor of epidemiology there. And he simply looked at, in his data sets, including UK Biobank, whether these hallmark-related diseases were associated with particular aspects of social deprivation. And what he found was that deprivation-related diseases are very strongly related to these aging hallmarks, much more so than ones that aren't. And again, it's the same cast of characters: stem cell exhaustion, nutrient sensing, intercellular communication, and mitochondrial dysfunction. These seem to be the ones that are really associated with social deprivation and therefore bring down the diseases that are associated with that mechanism. In some ways, I find that quite an optimistic bit of information because it implies that if you can interfere, you might particularly help people who are in socioeconomic bad circumstances, because they're the people who are getting these hallmark-related diseases. So what interventions have we got apart from the usual diet and exercise? I should say there's a great deal of snake oil associated with this area. It sort of started almost immediately in the 90s, once the papers on mechanisms of aging started to reappear. And one of the first appearances on this scene was this stuff, resveratrol, which is present in red wine. And there was an almost Ponzi scheme of papers talking about the wonders of this stuff. There's absolutely no solid evidence at all. And the same is true for many of the drugs that appear in drugstores and chemists on the supplement shelf. They haven't been through clinical trials. There is no evidence at all that they help. But it's a very, very profitable industry. Personally, I would never take anything that has not been through a clinical trial. And the latest development is these longevity clinics. I'm not sure if any of you have come across them. They started in the US. They're a real big business there. But they're appearing here now. They're basically for the wealthy, worried well, who can get everything measured, get told to eat a decent diet and take exercise, and pay a lot of money for it. And this actually appeared in The Guardian. I think it was last week. People are turning themselves into lab rats. There's an injectable peptides craze. Well, that's really dangerous. You could just go into anaphylactic shock. So, it's amazing what people will do to themselves. And there's now this idea of biohacking, where you do your own personal self-treatment, decide what's wrong with you, what you're going to have, and inject yourself with peptides. So there's an interesting fringe there. But there's also a lot of very sensible stuff going on. So these were the sponsors of the standard annual meeting, Aging Research and Drug Discovery. You can see the enormous number here. Some of them are doing in silico stuff. There's stuff on skin, and there's stuff on rejuvenation and pushing back biological age in the markers on the DNA. And a lot of this is actually financed by extremely wealthy people. So, for example, Altos is partly financed by Jeff Bezos, which has led to the idea in the field that we've got a new model organism, the billionaire. But there's a lot of activity. I'm particularly interested in the prospect for geroprotective drugs, either existing or new drugs. I know that people often, and perfectly reasonably, don't terribly like the idea of taking a drug for a disease that has not yet happened. But there's plenty of evidence that it works. I mean, the huge decrease in cardiovascular disease over the last decades has largely been down to drugs that lower blood pressure or statins that control the lipid profile in the blood. And it's been an incredibly successful campaign, and millions of people are taking these drugs. So there's plenty of precedence for that basic approach. And our own work's been concerned with this nutrient sensing network, the whole DAF2 story. So there it is in turquoise at the bottom of the wheel. This is just a picture of what it actually looks like in the fly. So we've got the receptor at the top here. So this is DAF2. And then a signaling cascade, sort of pile of dominoes that signals down into the cell. This is FOXO, I mentioned, the transcription factor that turns on stress resistance genes. And there's also this RAS pathway, terribly important in cancer. About a third of human cancers have a mutation in a RAS gene, and it's very much part of this network. And then also this TORC complex in the center here, which senses nutrients and stresses. So it's quite a complex network, and we're very interested in the idea of can we drug it to improve health, as are many other people. So two drugs here, rapamycin and trametinib. Rapamycin disrupts this TORC complex. Trametinib inhibits this MEK kinase here. So rapamycin is an interesting drug. It's a natural product. It was discovered on Easter Island with an expedition there that was looking for drugs that inhibited the growth and cell division of mammalian cells. It's actually used to suppress immunity at very high doses to stop kidney transplants being rejected. And it's also used as a cancer chemotherapeutic and to prevent restenosis after cardiac surgery. And interestingly, it turns out to be geroprotective. It's the drug that's furthest down that route at the moment. So it was the first drug that was clearly shown to extend lifespan in mice. This was in the US as part of the intervention testing program. And you've got the survival curves of females and males exposed to increasing doses of the drug. And you can see the very nice dose-dependent increase in lifespan. And it seems to interfere with multiple hallmarks of aging. It disrupts that TORC1 complex, but with consequences for several of the other hallmarks. And it's currently in several clinical trials. I mentioned this in the context of the gut structure and leakiness. But one of the published ones I think is very interesting. This is the work of Joan Manig. Older people respond poorly to immunization against flu. And because of the effect of rapamycin on age-related inflammation, Joan decided to look at whether it could improve the response to vaccination against flu in older people. So she didn't actually use rapamycin, she used proprietary drugs with the same effect, because this is part of a pharma program. So what she did was to take people who were about to be immunized, pre-treated them with her drugs, withdrew them and allowed them to clear them, and then immunized. And she found both that the direct immune response to the immunization was better, and also there were fewer viral infections in the ensuing winter. In fact, it looked as though not only did the drug potentiate the effect of the vaccine, but it was also having a direct effect on viral infections because they didn't only get less flu. So I think this was a very neat clinical trial saying that quite a short treatment with this drug can have an immediate benefit. It's actually a very interesting drug because it has really long-term effects. You can have a short treatment early in life in both flies and in mice and see an effect much later in life on health during aging. So it looks as though it can be used in short, sharp bursts, which is what you want for a drug like this. The other drug I showed you, the MEK inhibitor, trametinib, that's a standard pharma drug. It was developed by GSK to treat malignant melanoma. And we initially found that it was geroprotective in flies. It turns out also, we found recently that it's geroprotective in mice. So these are two different doses of the drug here in females on the left, males on the right. We were being super cautious with the amount that we used. I'm sure we can go higher than this, but you can see already we're seeing the increase in lifespan. And because of that complicated signaling network with the RAS pathway, the PI3 kinase pathway, TORC1, and so on, it's quite likely that this is a signaling network where actually more than one drug could be very useful because it would prevent the many feedback loops and also those transcription factors that the different branches talk to actually target slightly different sets of genes from each other. So we were able to look at the combination of rapamycin and trametinib in mice. And the results of that are shown here. And you can see that, you know, by mice standards, this is quite an impressive increase in lifespan in both sexes. So the orange curve here, the right-hand one, is the two drugs when they're administered together. And looking at we're still working on this project, but looking at what seemed to be causing it, there was a very strong reduction in age-related inflammation. So there was a clear immune effect, as turns out repeatedly with this drug, but also at least two organs show considerably reduced incidence of tumors. So it would be very nice now to be able to push this forward into clinical trials. This type of clinical trial in humans is not without its challenges. This is one that's been proposed in the US for a long time with metformin, which is the first line of defense against type 2 diabetes. It's an extremely safe drug, which is taken by very large numbers of people, and there are reasons for thinking it might well be geroprotective. And the important advance here was that the clinical indication was for aging. So this is the FDA in the US actually allowing aging to be an outcome of a clinical trial. What that means in practice is several diseases, but it's an advance to allow that. But the challenge of these trials is that even if you're repurposing an existing drug like rapamycin, it's off patent and it's cheap. So nobody's going to make any money out of it. And if you're starting to use it for a new indication, and especially if you're using it in older people, then it's going to have to go through clinical trials again. And if you're looking at multiple disease outcomes, it's going to be very expensive because you're going to need a lot of subjects. So really pushing this forward, I think, with all those kinds of challenges, the fact that you need long-term drug treatment, a lot of individuals, you have to go through safety again because it's a different target population. You know, the regulators are going to be very interested in that. And the pharmaceutical industry aren't going to touch it because they can't make any money out of it. So you're really looking at charities and governments for this. But on the other hand, it's clear that if we could get it through the valley of death, that the health economic benefits, even if you look at it in brute economic terms, would be enormous. And of course, in terms of individuals, it would be huge. So let's hope that is the promise and that we can get some of these drugs into clinical trials. So to finish, I think there's a real global challenge with reducing the burden of life diseases. Social deprivation is the elephant in the room when it comes to this. There's an opportunity to prevent multiple age-related conditions simultaneously by targeting these mechanisms of aging. Many existing drugs target mechanisms of aging and show promise for repurposing. And I hope one day we're going to see this broad spectrum preventative intervention for aging. So thank you very much for listening.
Okay, now we have some time for questions. And we have two microphones. Please don't ask a question without using the microphone, because that means that the people who are listening online can't hear what you say. So just put your hand up if you'd like to ask a question.
Thank you very much. Great talk. From an evolutionary point of view, animals, life has been subject to toxins and has presumably in the environment and maybe from its own internal mechanisms for, you know, that's the way it is, and have developed mechanisms for dealing with them. We are in a new world with all sorts of new toxins. Can you say something about that and how that could affect aging?
Yes. So as far as the evolutionary side of the question goes, there's quite a lot of evidence that, for instance, FOXO, that transcription factor, part of the cast of characters that it activates is actually the detox pathways, the standard, you know, phase 1 to phase 2. So it seems clear that in defending cells, defending cells during aging, that whole process is very important. As far as the modern environmental pollutants go, there's been very little work on it so far, partly because I think sometimes we just don't know what's going on. So, there's this rise, this is not an aging related thing, it's the opposite. There's this rise in colorectal cancer in the younger section of the population. But I think there's a strong suspicion that that has to be some sort of environmental pollutant, but nobody's got any idea what it is. And we can certainly use some of the ones that we know about and start to look at their effects on, organoids and whole organisms. But I would say that whole line of inquiry, as far as aging goes, is really in its infancy. But I think it's really important. Actually, quiet audience.
Richard, Adam wants to ask a question.
Thank you kindly. It's a bit weird being on this side. My understanding of aging is that it primarily connects to telomeres, I believe it's pronounced, as in the protective caps of the chromosomes. Now, feel free to say if this is going more into the side of snake oil. My understanding is that this is affected by free radicals and it's to do with oxidation and all this jazz. Could you weigh in on that by chance?
I'm sorry, I didn't hear.
Mhm. For some reason, Adam, it's quite tricky to hear your question. Do you want to come down and ask it from the microphone at the front?
Yeah, I'm really sorry, I didn't hear.
Is this any better? No? Yes?
Yes, that is.
My question was to do with telomeres. I know how to use my microphone. Thank you. My question was to do with the protective caps of chromosomes, i.e. telomeres. My understanding is that as we age, they get more and more damaged. From there, this is going maybe more into the snake oil idea. Isn't it that as we get older, there's more free radicals, there's oxidation and all that jazz? Could you weigh in on that?
I mean, there's certain telomere, sorry. There's certainly telomere erosion, particularly in types of cells that divide during aging. And it's a very important cause of cellular senescence, because once the telomere is eroded to the point where it's starting actually to nibble into the chromosome itself and the genes, then you get the cell tipping over into cellular senescence. And then you start to get the tissue damage. And there's a lot of experimental evidence now that if you remove senescent cells from tissues, this is in mice now, either genetically or with a drug, then you improve the tissue health, and you can also actually make the mouse live longer. So it's very clear that the ones that accumulate during aging are damaging. One size doesn't fit all. Cellular senescence is very important in wound healing. So the real role of these cells during development and wound healing is that they appear and they remodel the tissue. That's why they can destroy tissue, because often to remodel it and heal a wound, you need to kill bits as well as make bits. But the problem is they appear during aging, and normally when they're functional, they're removed by the immune system. But for some reason, these ones that appear during aging don't get removed, probably because of some sort of immune deficit. And so they accumulate, and definitely telomere erosion feeds into that.
I'd like to ask you a question, Linda, in connection with the graph you showed at the beginning of your talk, where you mentioned that women live longer than men, but their healthy lifespan is shorter. I wondered to what extent the menopause in women might play a role in that deteriorating health without shortening lifespan. Do we know that, I mean, we know the menopause doesn't do you any good. Is it the case that the kind of damage we associate with the menopause is not actually linked to lifespan?
I'm not sure if we know, because we'd need to have individuals who didn't go through the menopause. You'd need to have some sort of comparison. But certainly the sorts of things that are associated with menopause, I mean, osteoporosis, muscle weakness, these are all bad things that happen during aging. So certainly some aspects of age-related ill health in women are related to menopause.
So you might then think it would reduce lifespan in women, but they live longer.
Yes. So I don't think anybody knows I mean, there are about 5 different hypotheses to explain that difference. And I don't think anybody really knows, you know, what the right ones are. There's one interesting bit of biology, which is that there are some organisms, I mean, humans are one of them, with a long post-reproductive lifespan in females. And there's this idea of the grandmother effect, that actually it's an advantage to have your grandmother around. And in hunter-gatherer societies in humans, which is what we've evolved from, there's some evidence that that's the case. Actually having grandma around can either increase the reproductive success of daughters or the survival of grandchildren. But a particularly clear case is actually killer whales. Killer whale females have a very long post-reproductive lifespan. And it's absolutely clear from the numbers that if you've got a post-reproductive grandmother in the pod, the young males survive much better than if she's not there. So there may be advantages to a post-reproductive lifespan under certain restricted circumstances, but certainly the menopause is not good news for overall health.
Does reducing inflammation increase lifespan?
Sorry, does what?
You think inflammation? Inflammation.
Inflammation, yes. Yes, there's plenty of evidence for that in experimental studies with the model organisms. I mean, this age-related inflammation is not because they're becoming more infected. There's nothing adaptive about it. And if you tamp it down, I mean, including with rapamycin, it's clear that it's advantageous. Yes, it's a negative feature of aging. And it's accompanied, and it seems it may even sometimes be causal in making the immune system work less well in a functional context. So it damages the system itself. And a lot of it is to do with immune cells getting into the wrong places. So for instance, one of the reasons that obesity is so dangerous is that the fat becomes invaded by inflammatory macrophages and then starts releasing lipids into the bloodstream. And that's very damaging. So yes, it's definitely a good idea to tamp it down.
I wonder if there's possibility that doing that application of the disturb other mechanisms which throughout effort are these drugs which have been well understood that the risk of that.
That's an incredibly important question. I think a lot of the question mark over that is to do with dosing. So the doses of rapamycin and trametinib that are used in the mouse studies are a lot lower than the doses that would be used clinically. And the dose of her proprietary drugs that Joan used in that immunization project induced no side effects at all. She was working at doses way, way below what you'd use as a cancer chemotherapeutic or to prevent kidney rejection, where you do get quite major side effects. But I think what we do not know for any of or either of those drugs at the moment is what the consequences of taking them for a long period are. We don't know about, you know, chronic treatment and the kind of side effects that might appear with long-term use. That's why I think the fact that rapamycin can be used in very short bursts is really important. The metformin trial, I think, would have been very interesting. And if they can get the finance for it, I think it will be really important because many people have taken metformin for very long periods. And it clearly is a very safe drug with long-term chronic treatment. So I think it's a prime candidate, in a sense, for the first long-term clinical trial in humans because we already know how safe it is. But the question of side effects is obviously crucial.
Your mention of obesity reminds me that I was at a talk recently by Navid Sattar showing the effects of weight loss drugs that are actually reducing, protecting against a whole range of what would be age-related diseases. Is that another promising pathway for?
It's absolutely clear that that you could almost describe them as geroprotectors. I mean, they've only been around for such a short time that we can't possibly know. I mean, nobody's taken them for long enough to know what the consequences there are. But I find it really easy to believe that they would be very protective during aging, simply because of what we know about the socioeconomic deprivation story and the hallmarks. So you can go on from those I didn't have time to mention it, but from those epidemiological studies, we know that almost any measure that you use of social deprivation is associated with acceleration of the hallmark-related diseases. And if you go on and say, okay, let's look at the characteristics, other characteristics of the individuals in those studies, which ones are associated with the acceleration? Number one is obesity. Comes straight out. Number two is current smoking. Number three is low level of education. But education is almost certainly acting as a mark for health-seeking behaviors, other things. But obesity is always top of the list. So if you can reduce that, you're going to reduce a lot of age-related diseases.
Hi, thank you. Great talk, by the way. You mentioned a lot of interesting experiments on the effects of drugs on mice and expanding the healthy lifespan. And I guess that's good because mice don't typically live very long, so those studies can complete quite quickly. But in terms of moving this on to a human clinical trial, my assumption is that we'd need to wait for all of the humans in your clinical trials to pass away to assess the effectiveness of those drugs in humans. And so I guess I'm speaking a bit more practically in that, assuming these clinical trials in humans are successful, how soon could we have these drugs be available for humans on a commercial basis? And secondly, speaking as someone who's relatively young, at 27 years old, do you think those drugs will be available for me in time?
I'm going deaf. I didn't hear the last bit. What was it?
You're only 27, he's wondering if he should start.
Right. Yes. So what was the first bit of your question? Sorry, there were three bits. When should you start? I think we'd all hope that you wouldn't have to start at least till middle age. I mean, a lot of these things that go wrong during aging are basically life course effects. I mean, it's clear that even effects in utero can be important on how people have an important effect on how people age later on. And some of them at the moment, we don't know how to reverse. It would be nice to think it's never too late, but probably it sometimes is a bit too late. But ideally, what we'd like is interventions that we could make start in late middle age and would be beneficial from then on. But I think for a lot of these, we don't actually know. And I remember the first bit of your question now. You're right about the long-term nature of the clinical trials in humans. One of the things that none of the regulators will allow at the moment, and I think there are good reasons, but it may change, is the use of biomarkers rather than a disease outcome. But if we can do enough work with the model organisms, that we've got a clear indicator, a molecular indicator, that we're hitting the right target and that therefore a particular disease is less likely to occur. It is possible, I think, that in the future, we'll be able to use predictors in clinical trials rather than having to wait for the final outcome. But at the moment, if you talk to any of the regulators, they don't want anything to do with it. And I think that's absolutely fair because I don't think the biomarkers are accurate enough at the moment. But I think they will get more accurate. I mean, that's the huge benefit of these long-term studies where you collect samples and plasma and so on all the whole way through. And you can then see what happens to the person, but go back and looked at whether you could have predicted it when they were young. So there's a lot of that going on at the moment.
Hi, thank you. I just wondered that when you mentioned these promising developments, which sometimes companies won't look at because there's no money in it. And I just wondered, is there any campaigning organization or is there any agency that's trying to work at developing more integrity in the pharmaceutical world?
That's a very interesting question. And it's something we have. It's something we have a lot of discussions about. Is how to deal with the lunatic fringe. So, you know, they come together at that Copenhagen meeting, which incidentally has been cancelled this year by the university. So watch this spot. But that's where there's a lot of serious science, but also some more dubious stuff, and who is actually financing it. It's where, you know, the whole boundary of what should and shouldn't happen sits. And I think how you actually negotiate that in terms of trying to, you know, keep what people do as respectable as possible and make absolutely clear the distinction between scientifically rigorous work and puff and things that have been through a clinical trial and things that haven't. I think it's something the whole field actually needs to pay a lot more attention to and be a lot clearer about.
First of all, thanks very much for the discussion. Following on from the last question, you were somewhat dismissive of supplements, etc., on the grounds that there had been no trials carried out to prove one way or the other whether they work. But later on in the program, you mentioned the same truth about rapamycin and the trials for the combination, which seems to fall on the basis that as it's out of patent, no pharmaceutical company can make money from it. Therefore, there are no trials done. So I'm wondering just if you can give me a broad idea of what one of these clinical trials costs. So we might maybe approach Mr. Bezos to put his hand in his pocket.
Yeah, that's a good idea. I mean, you're up in the hundreds of millions, because if you've got to follow people, say, for 10 years, and you're looking at a 5-disease outcome, and you're looking at men and women, and you've got the right ethnic mix in there, so the whole thing's stratified according to the variables that you know are going to be important in the way that people respond to a drug, you're looking at a very, very expensive trial, even by clinical trial standards. Well, I think hundreds of millions is quite expensive. No, you're talking seriously expensive. Bezos money.
Well, it's said that 55 years old is old in Bridgeton, whereas there are many people in Bearsden who are 75 years young. The difference between biological and physiological aging. And I wonder to what extent the literature you describe spans both aspects of aging. I'm thinking in particular that participation in trials and research is much less in deprived areas. And you mentioned Biobank, which recruited 5% of the population. And I'm just wondering whether there's a gap there.
Yes, I mean, certainly there is. Who volunteers for a long-term study, the longitudinal cohorts, that's not going to be a random section of humanity by any means. It's one of the reasons I particularly like the health record, because there are some people who would never go near a doctor. I mean, there is still, a non-random aspect to it, but you've got a much better, I think, cross-section of the population if you're looking at GP and hospital records than if you're looking at people who volunteered to take part in a long-term study. So yeah, I think the issue that you raise is a very real one, and it's one that increasingly people are starting to address in clinical trials. I mean, there's been a very serious bias there in the past. They've almost all been done with young men. So, you know, there is a problem. And increasingly when people look, you know, these other variables, age, gender, sex, and so on, do affect how people respond to drugs.
I've been diabetic 2 for 25 years. I take metformin, etc., simvastatin. Last year, I was diagnosed with prostate cancer, and just got the result a few days ago that I'm clear now. I went through radiotherapy and the other treatment, you know, so I'm still here. And can you give me any the recommendation? I'll be 76. I'll be 76 in July, but sometimes I feel 26, you know.
You see, it's what I was saying about Billy Connolly, the yin and yang of, you know, there are these beastly diseases, but there's also healthy aging going on in parallel. So let's get rid of the diseases.
On top of the radiotherapy, but hormone treatment as well. That's to calm down the testosterone.
Yeah.
Anyway,
God bless the medical.
I feel good, and I just thought I want the world to know.
Right.
Thank you very much for your very, very important talk. I just wonder whether, in your view, is it at all possible that by healing, people in old age can recover from diseases? Because there are instances that they do report that somebody without any surgical operation has been cured or healed.
I'm not sure I really have any wise words about that.
Well, I mean that with some people may stop all the diseases associated with old age as a result of religious belief or spiritual dimensions of their lives, or healing as it is operated and practiced in many religions.
Well, I mean, it's just to some extent believable because, I mean, all of these things, if they have any effect at all, are going to be doing it through biological mechanisms. And it's clear, for instance, that certain kinds of stress are really bad for you. Not all, but some. Not sleeping properly is really bad for you. So if it's feeding into those kinds of things, then it could be having an effect.
Just like to add that I do doctor.
Very wise.
We've got time for one final question. Is there one over there? Yes.
Thank you. It used to be the case that full dental clearance and false teeth were a common 21st birthday present in Glasgow. Dental caries is a potent source of inflammation, chronic inflammation. It's the most common cause of surgery, I think, hospital admissions in the under 10s, and it's closely related to social deprivation. So I've got two questions to you. How many times a day should I brush my teeth? And has there been any strong data linkage between dental health? I don't know if the English, they can actually, can they link up dental records and GP records?
I think you've raised something really important. Dental health is so important, not just for the teeth and the mouth, but, you know, endocarditis, for instance, with, you know, infections. And gum disease is something that in mice is actually reversed by rapamycin. It has a really dramatic effect. But what infuriates me about the whole thing is, first of all, the whole situation with national health, or the lack of situation with national health dentistry. I mean, the standards of dental care throughout the British Isles, I think, are absolutely appalling. And actually, I have occasional fights with my own dentist about it and say, why on earth don't you guys get involved in more clinical work? Why aren't there better records? You know, why don't you get yourselves organized so that we actually have a picture of what's going on with people's teeth nationally and how it's affecting their health? They just seem to be not involved in research. Sorry if I'm offending any dentists in the room, but I do find it quite extraordinary that there isn't more clinician science going on in dentistry.
Well, perhaps on that not so happy note, especially given that the west of Scotland has traditionally had a lot of poor dental health, we should draw this to a close. Just one final thing I'd like to say. I won't ask Linda to comment on it, but there is data showing that baldness in men is not linked to lifespan. I say that looking around the room hesitantly. Nor is graying of the hair. So we're all... Anyway, I'd just like to thank Linda so much for a very wide-ranging and informative talk on something that is very relevant to society. You probably know the Scottish Government produced a report just in January on healthy aging and what society needs to do to try to get it to happen. And research of the kind that Linda's doing is very, very important in this context. Whether we're all looking forward to taking a pill or not remains to be seen. But just before then we thank Linda collectively, there's two things I have to do. Actually, maybe three things. I'll just remind those that were getting the chocolate that chocolate is these days considered to be good for you, so don't forget to pick up your chocolates. It's my pleasure on behalf of the Society to present Linda with the Society Graham Medal in honour of Thomas Graham, the chemist, who was very influential in our society and, of course, in the discovery of dialysis. And then, secondly, to present her, of course, with the Royal Philosophical Society paperweight.
It's a really beautiful medal. Thank you so much, Pat. This is a real honor.
The really beautiful paperweight.